So, PDB is the database. It's a primary database. It contains annotation. Some amount of annotation can be found in PDB. We can search as RCSB PDB. This Royal Society is been maintaining this website RCSB. So, PDB means protein data bank. So, the data of mainly structure of protein, as they have structure of protein, with the structure they have the sequence of protein also. For with every structure they have the sequence of proteins also. So, that is the protein data bank, and this RCSB protein data bank, if you want to know more about the RCSB protein data bank, you can check this website, PDB 101. This is a tutorial website which will give you a lot of tutorial on how to use PDB and all, how to use PDB, how we can download files, all things related to PDB can be seen, tutorials can be found in the PDB 101 website. And PDB is a part of WWPDB consortia, just like we can say International Nucleotide Sequence Consortia, where the NCBI, EBI, DDBJ combines together to form the International Nucleotide Sequence Consortium. Just like that, WWPDB, this is a inside this consortia, this RCSP PDB come, and rest of many other websites also come. If you go WWPDB, you can see the websites, crystallographic associations, or many websites have been or many databases has been involved in this consortia. And seeing the PDB, someone is there. So, seeing PDB website, PDB consists of 2 lakhs structures in PDB. Mainly the PDB structures are derived from either by X-ray crystallography, NMR spectroscopy, that were the most widely used techniques earlier. So, now as this cryo electron microscopy came, mostly the determining structure of the cryo electron microscopy is become more faster compared to that of the earlier X-ray crystallography and NMR spectroscopy. Nowadays more structures are coming from cryo electron microscopy. So, actually the PDB, these 2 lakh structures include the structures which derived from X-ray crystallography, NMR spectroscopy and cryo electron microscopy, and they have we are going to computationally model a protein. So, like that modeled protein also present in PDB, it's around more than 10 lakhs modeled proteins also, computer generated proteins also is present in PDB. So, when we see the sequence, we can see this symbol, this symbol is experimentally determined. If this symbol come aside of the sequence, we can see it is experimentally determined structure, or if this computer sign come, we can say that they are computer determined structure. So, like that, this is the PDB. PDB usually select one molecule as the molecule of the month every month. So, then we can search our proteins in the PDB, so that we can get the structure of that protein. I will first search a spike protein, which was in story earlier. I will select a SARS-CoV spike protein. It belongs to this spike protein is determined by cryo-electron microscopy, and this SARS-CoV means Schwinn acute diarrhea syndrome coronavirus. It is not a SARS, so it is a different virus, and it's a spike protein we can see here. So, these are the authors of that protein, and they have determined the structure in 2020, and this is spike glycoprotein, and their ligand present they have said. So, we can click the protein and we can see the symbol here, that is the conical flask, So it is experimentally determined structure. It is determined by this cryo-electron microscopy, and it has a PDB ID, just like a accession number in the case of GenBank. It has a PDB ID. I think today's class will be bit more. I am trying to tell everything about the three techniques, and if it go further, please be there, and I will be completing all the three techniques, and I can't make it for tomorrow. Okay, so please be there. Otherwise also I will continue. If you people are not there also, I will continue and send you the recordings, because after as the class is going to close, we are having very less participants. Many of them leave in between. So I am clicking this cryo-electron microscope structure. So we will get the cryo-electron microscope structure and its details in the PDB website. You can see this digital object identifier DOI number is there. Permanent URL is the DOI number, and it's the EMDB map, and then the classification is a viral protein, and organism, which organism's protein is this, and which expression vector is used to clone that this spike protein, and further cloning and sequencing, which vector is used in the process for cloning, and when it is deposited, and the funding organization, it is from China. They have only studied and submitted this, and this region is about experimental data, what all parameters they have used for the experiment, and they have used electron microscopy, and the resolution of the structure is like this, and so the various parameters of the parameters they have used in the electron microscopy, cryo-electron microscopy, they are mentioned here, and this is these things are about the quality of the particular structure, which was denoted by the class score, Ramachandran outlays and side chain outlays, which gives the quality of the particular structure, and the structure is published here in 2000, when it is published 2020, in which journal, everything you can see by going to this DOI, and it contains some amount of annotations also. You can see glycoprotein, it contains chains A, B, C, sequence length, organism, mutations, all these things you can see here, gene name, what is the gene name, and link to various other databases, Uniprot and hydropathy, it's hydropathy-prot, which of the residues are hydrophobic, which of the residues are hydrophilic, and disorders in the structure, as well as PFAM, which family it belongs based on the domains and motifs. So all these features are links to the other database and tools we can see in this page, and the structure weight, you can see how much the weight of the structure and how many atoms are present. So modeled residue count, so how many atoms are present here, and how many amino acid residues are present. Atoms is about 22,000 atoms are present in this molecule, and 2,811 amino acids are present in this molecule. So while clicking this download file, you can download the faster sequence of this SARS-CoV spike protein. For if you need for some analysis, you can download a faster sequence, and PDBX-MCIF, that is the format which is PDB is been researching. This is going to be the future format for PDB. We are not downloading that. They are doing lot of adding more data to this PDB-CIF format. So we will download the PDB format now, and while downloading the PDB format, this PDB format, downloaded PDB format can be used for visualization, or can be used for analysis, etc. and we can see the actually earlier the PDB was classified as a primary database. Now it contains lot of annotation. We can say it is a secondary database. So you can see the structure of this protein, and you can click anywhere so that we can see it elaborate, and we can see that amino acid in the sequence also we can see. I need to find it out. It will be marked in the particular. So we can see, we can closely we can see what amino acid is present here in the particular chain, all which can be seen in this, and it will need to be denoted here also to find that. You can see which you can see where this we have selected the tryptophan 735 tryptophan only we selected. So I think 735, where is 735? I think here, so it is not shown. So it's not shown, but here we can see the tryptophan 735 position tryptophan is what we have selected. So we can select the amino acid. You can see 309 734, like that a different amino acid we can select and see the various features of the amino acids here. And annotations we can see about the protein. You can see the proteins various annotations, domain annotation, various domains present, and their function, their homology, all these things, annotations of the various domains present in the particular spike protein, and its 14 which family it is present, and their description, their details of that receptor, and the gene product annotation, what is the gene product, all these annotations are present in the annotation page. And experiment page is very important when you are doing the cryo-electron microscopy of these or related proteins. You can follow these experimental details in your machine so that you can get the structure properly. So the experimental details, what all things, what machines they have used, and what all parameters they have used. I think they use this microscope, this cryo-electron microscope, FEI Titan Cryos. So which microscope they used, and which imaging mode, all these things, everything about what all experiment they have done to determine the structure will be seen in the PDB database. You can use that data, that thing for your experiment. If you are working on that, you can use it for that. So sequence of that particular spike protein can be seen here, and this sequence can be downloaded as FASTA format, and sequence at various sequence means its various properties at various database we can see its second structural elements we can see here. Where is helix, where is sheets, we can see when I am going through, we can see where is helix, where is sheets, and various functions of the protein depending on the database contain data, that all been included in this chart. We can see what disorders are present here. You can see disordered region, and this is you can click there and get the idea of that where disordered the protein is disordered or not. So like that, hydropathy, which residue is hydrophobic, which residue is hydrophilic, all we can find out using PFAM, protein which protein family. You can see beta coronavirus like spike like a protein. You can see in the top, when we click there, we can see in the top about the features of that particular protein. And then there is a genome page, and then the version page also is there. There is no genome alignment is available for this sequence. So like that we can see the structure here, and we can download the PDB format for visualization. We can visualize it through, I have downloaded the Rasmol. So, if I click here, it will be directly open in Rasmol. So, actually what is inside this PDB file, that you need to understand. So, it will be directly open in this PDB file will be read by the molecular visualization tool, and it is going to open in, I think it is going to open in Discovery Studio. Rasmol also is, I have, and not selected. I have both tool. So, let us see anyway if it is Discovery Studio or any other tool we can see. Let it open. So, actually this Discovery Studio will read our file, PDB file, and it will visualize the structure. Yes, we can see the structure here. We can see the structure here. So, we can see the structure here. We can rotate and see we can see some areas are contain the helix, some areas contain the beta plated sheets, and loops we can see here, and many other ligands are attached to the this protein can also be visualized here. So, you can see the protein, spike protein clearly by using this any visualization software. I have Rasmol also, I thought it will open in Rasmol. So, it is a Discovery Studio, another tool which is used for the visualization. So, it is open in that. So, like this we can see, and so that is the thing. So, we can see that actually this PDB file which we downloaded, we can see the PDB file, actually what it is, what it codes for, or what is present in this PDB file. This usually this PDB file is like this. This PDB file will be read by the this PDB file will be read by this tool, and they convert into this structure. So, the tool will read the PDB file and convert that PDB file into this structure. Visualization tool. And what is present in this PDB file? We are usually going through every files because we have gone through GenBank file, we have gone through FASTA file. We usually study all the files because we need to understand what is present in the PDB file. Usually the PDB file consists of it lists out all atoms present in that protein. Okay, list out all atoms and their distribution in x, y, and z axis, that is a 3D. So, in the 3D plane, how these atoms are being distributed, which finally result in the structure of the whole structure of the particular protein. Okay. So, the PDB format contain x, y, and z coordinates of a particular atom. These are determined using X-ray crystallography, NMR spectroscopy, and cryo-electron microscopy. So, we can see this is the first atom. This is associated with the first amino acid histidine. So, this histidine amino acid is associated, or histidine amino acid is made up of you can see how many atoms? 10 atoms. So, the first amino acid is histidine. The histidine contains, is made up of 10 atoms. So, these are the data present. Okay, these are the histidine is the first amino acid, which consists of 10 atoms. These are the atoms: nitrogen, this carbon, oxygen, carbon. These are the atoms. These are the atoms listed. Atom names listed. So, histidine is the first amino acid present in the protein, and it consists of 10 atoms, and the atom name is listed here, and the number of the amino acid, which number is listed here, and here the x, y and z, the distribution of these atoms in x, y and z plane is also listed. These are the data mainly present in the PDB. Some more data are present. We have not discussed about this fourth line. This fourth line mainly is a chain identifier. So next we'll data field residue, and notice that 2 like residue adjacent to next data field is XY. Okay, this is a chain identifier. This A, B are the chain identifier. First residue in the chain is histidine, and second residue in the next field data is a residue type, that is amino acid, and next data is the chain identifier, which chain the particular amino acid is belong. So the A chain we can see here, which chain, this chain identifier, this is chain identifier, which chain the particular amino acid belongs to, and the last 3 things what it is. So last 3 things are last 3 things, last 3 field are first is the occupancy, second is the temperature factor or B factor, and last is the element symbol, which is the element occupied in the particular position. Element symbol only we have seen in the last, and this is occupancy and B factor. So B factor. So occupancy and B factor. Occupancy and B factor, I will tell what it is. Okay. Rest of the things I think you hope understood. The PDB file contains the list of atoms and the distribution in X, Y and Z planes, so that to get the 3D structure of the particular protein. Okay. And the atoms are numbered in the second row. Atoms are numbered, and in the third one the atoms name is being indicated, and fourth one where this, to which amino acid these atoms belong. That is here histidine and serine, we can see down, and which chain the particular atom is present, and the next one is which number of amino acid, first number, second number. Serine is a second number amino acid. So I think glutamine is the third number of amino acid. So which number of amino acid, and this is the element name, and these are the occupancy and the B factor. Occupancy and B factor. What is occupancy and what is B factor? That's the only thing left in the PDB file. This PDB file is read by any of the molecular visualization tools, such as the RASMOL. You can easily download. All the tools which I am showing here in this workshop are freely available. You can easily download. You can just write the names and you can easily download from freely from the internet. Some of the software is very big, so it's not running properly in my computer. Anyway, I will try to show you as much good as possible. So occupancy and temperature factor, that is the B factor. Occupancy is the proportion of time that an atom spends in occupying a particular position. The field is especially important when a molecular region is flexible. If a molecular region or some regions of amino acids are flexible, is it? Some regions, the psi and phi angles, the Ramachandran plot you have seen, some regions of the amino acids are flexible. So some if the atom present in that region of the amino acid, we can the occupancy tell about the proportion of time that atoms spend in occupying a particular position. So the proportion of time the atom spent in occupying a particular position, it is called the occupancy. So occupancy is then this line, we can see the occupancy, the proportion of time the atom spend in a particular position is called the occupancy. And B factor. B factor can be indicating the relative vibrational motion in different parts of the structure. Atoms with a low B factor belongs to the part of structure is well ordered. Atoms with a large B factor, which is generally belongs to the part of which are very flexible. So these also both two factors tell about the how whether the atom is flexible or ordered. So the what occupancy tells about the proportion of time atom spend in occupying a particular position, that is occupancy. B factor tells that the relative vibrational motion in different parts of the structure. The lower the B factor, that part of the structure will be ordered. So we can see here B factor is less. So this part is less ordered compared to this part. Lower the B factor, that region is very good ordered. It is not flexible. It is very well ordered. Higher the B factor, that region is very flexible. It is not ordered. So B factor and this occupancy tells about the flexibility or orderness of the atoms or orderness of the structures present in a particular protein, or the orderness of the parts of a particular protein, parts of the structure or orderness of the atoms present in amino acids in a particular protein. How it is is there any vibrational motion occurring within in the atoms, so that the structure become more unstable or flexible, or the structure is just highly ordered with a low B factor. So these all things say about the say in the PDB format. This format file, now it is the Discovery Studio or else the Rasmol, all this take this file and they convert it into that structure, 3D structure, and we have seen in the this Discovery Studio. So this is all about the PDB file. Can see the PDB file. So some of the PDB files you can see, we can take another protein, hexokinase. This is usually this structures has been derived from this electron microscopy, contain less data only, very less data are only seen. So you can see if it is usually structure is determined by this X-ray diffraction, some values we can see, R value free, R value work, R value observed, like that. These values are these are the experimental values. What these values indicate, I will just go through and go through the next topic. So R value indicates the quality of the atomic model obtained from the, it's mainly seen, it is seen in the X-ray crystallographic data only. Usually in the cryo-electron microscopy structures we can't see that R value. Usually in X-ray crystallography, most structures present in the PDB are from X-ray crystallographic data. So the R value is the value which says about the quality of the atomic model, which is seen in the PDB. So we can say that R value usually it is typically 0.20. The perfect R value will be 0 for a perfect structure. So R value which indicates the quality of the atomic model, and the perfect R value or for a perfect structure will be 0, and usually the typical values will be 0.8, and so that is about. So depending on that we can determine whether the structure is perfect or not. So there are several reasons. You can go through the PPT. Several reasons why the fit or the structure is not perfect. One of the reason is protein and nucleic acid crystal, mainly based on these are technical things. Usually determine or generating the crystals for the X-ray crystallography is a tedious process. So during that several things can occur which can affect the R value. You can see the protein nucleic acid crystal contain large channels of water. Water does not have a defined structure and not included in the atomic model. Other reasons, so this water can result in some sort of this decrease in R value or some sort. It can affect the quality of the structure. So many other factors can affect the quality of the structure. Some other things come in the VDB are the clash score. You can see clash score, Ramachandran outliers, side chain outliers, RSRZ outliers. Mainly these are all seen in the case of X-ray crystallographic structures only. Mainly we have not seen in the cryo-electron microscope structures. So clash score is the score derived from the number of pairs of atoms in the model that is usually close to each other. So it is clash scores can be derived from the number of pairs of atoms in the model which are closely unusually close to each other. If clash score is the score which determine the number of pairs of atoms in that model which are unusually close to each other. So that is the clash score. Ramachandran outliers mainly are those amino acids with non-favorable dihedral angles, which tells about the amino acids with non-favorable psi and phi angles. RSRZ value is calculated only for standard amino acid and nucleotide in the protein, DNA, RNA chain. A residue is considered as an RSRZ outliers if its RSRZ value is greater than 2. If it is greater than 2, we can consider that residue is RSRZ outlier. So these are some of the values which can be used as to find out the quality of the structures derived using X-ray crystallography. One is the R value, which should be around 0 or typical value 0.2. And then clash score, which gives how much amino acid pairs which are come close to each other, unusually close to each other. So Ramachandran outliers will tell about the amino acids in that structure, in that model, which are having non-favorable dihedral angles, psi and phi angles. RSRZ value which tells about the usually the RSRZ value for a RSRZ outlier will be greater than 2. So RSRZ score compares the fit of electron density of each residue with the mean and standard deviation of the fit of all residues in the similar resolution bin. That is the RSRZ value. So the fit of RSRZ value compares the fit to electron density of each residue. The electron density of each residue with the mean and standard deviation, the fit to electron density of each residue with the mean and standard deviation to the fit to all residues in the similar resolution bin. That is the RSRZ value. So these are some of the things you should think when you are going through the PDB.